Hi everyone. Am I working? Can you hear me in the back? Excellent. Those of you who know me, you know my voice isn't very loud, and so people in the back, when my voice starts to fade, give me a boost there. It's really an honor to be here, and to be here with Alan, longtime collaborator, friend, and we never envisioned that we would in 2026 be in Madison, basically talking about this research based on our original conversation. So what we talked about today was to try to together tell the story of kind of how we sort of got here and kind of pass this on to you.
Not that we're going anywhere,
But you never know. And so we want to sort of pass it to you. And so what we hope you will do today, while we're here, while we're talking, and in our discussions, both formal and informal, is get everything you can out of us that you think might be worthwhile for you. No questions are silly. Just ask, right?
absolutely.
Yeah, let's start the discussion.
Tell us your name and ask us whatever you want, or tell us whatever you want. We'll be available.
So we're going to start in just a minute. We're going to kind of go through discovery, replication, exploration, and then some transitions, not only from us as people who are did some of the early work in this area, but other transitions that we think should happen. Before we start the formal presentation, I just want to say for all the people who feel like they're early in their career, whatever your formal label is, this is the field where you can actually make really important discoveries. It's not like this has been all picked over, 80% of the stuff's been already figured out. This one is wide open. So if you decide to make this a career focus, you can have big impact quickly. If you are rigorous with your science, careful with the questions you decide to ask, especially connecting with somebody who knows something about the clinical impact of what you're doing. You may not win the Nobel Prize by next year, but you got to start planting those seeds early. And so there is just so much work still to be done here. We have some experienced researchers here who've done that, who've really begun to focus on very important areas and are recognized within urology, both benign and oncologic urology, as movers and shakers. It's really good to see you here. So I'll start. Okay, do you want me to move the slides for a little bit? Yeah. Alrighty. Not sure this one's working.
There is a mouse on the desk if you just want to use
I got it. So this is a picture of me and Alan hanging out in San Diego. This is a favorite. This is where I live and work now. I am in my bonus career. And so one of the things I wanted to talk about is Alan and I have had fun doing this work. We worked hard. We worked really hard. We overcame a lot of barriers, but we had a lot of fun. And I'm trying to think if we've ever had an argument. I don't think so. If it was, we might have had a disagreement about who got to have the coffee first, whatever. Nothing serious. We really have been blessed with a very collaborative spirit and the joy of discovery and the joy of seeing the impact of the work be moved out into the scientific and even clinical fields has really sustained us and refueled our desire to keep going. Would you agree?
Absolutely. Yeah, I think that I told Linda a little while ago, looking around the room, the average age of the audience is well below 30, which means that you don't know the story. So we're going to tell, we weren't planning on this, but we're going to tell the story. But until a year ago, I had a departmental chair named by the name of Catherine Knight since the very beginning of my career. So 36 years she was my chair. And I have a tremendous amount of respect for the woman. She's very well put together. she told me, knowing that I was looking for something to extend my career beyond the circuit board inside of E. coli K12, which is what I was known for, I knew that the shelf life at NIH was pretty short on that, and I was looking for the right story to work on, the right person to work on. And Linda and Catherine had co-chaired some committee, and Catherine said you should introduce yourself to Linda Brubaker. So there was some sort of internal retreat on campus, something about sharing data, and we were both there. So I walked up to Linda and said, hi, my name is Alan Wolfe. We have 2 words in common, E. coli. You can work on it if you want to kill it. She said, let's talk. So Linda's very, very respectful of time, other people's time. So she set up a series of 1-hour conversations that started on time and ended in an hour. And so during one of those 1-hour conversations that we were just sort of trying to get to know each other and figure out what was it that we could do together. And she said, you know, some people think that the bladder is sterile. And I said, no flipping way, have you seen the plumbing? And then we spent a bit of time fumbling around trying to figure out, well, how do you test the dogma that the bladder is sterile in the absence of infection? And it dawned on me it was 16S5 gene sequencing. Next generation sequencing was brand new, and it was just becoming cheap enough that we could use a $25,000 grateful patient donation. But we needed somebody who knew how to do it, because I'm an E. coli geneticist and physiologist. I can extract DNA out of E. coli, but the rest of the stuff? So I was giving a seminar down in Bloomington, and the last person on my office visit list, the dance card, was a guy by the name of David Nelson, who was a brand new assistant professor. Listen, Michael, he was a brand new assistant professor. And somebody told me what he was working on. And so I told him that I was trying to work on sort of the same thing. And he spun around in his chair and started typing away on his computer, which is really weird. I don't know if you've ever had that happen when you sit down with somebody and they turn their back on you. Then he spun around and said, I just sent you a grant proposal. Use whatever you want. So ultimately, it was his postdoc, his wife, who did the extractions. It was the IU Bloomington Genomics facility that did the sequencing. And then David said, I know a bioinformatician. He's down at the University of North Texas. His name is Chuck Von. And he did all the bioinformatics. And then I got to play postdoc because I didn't have anyone in my lab that did this kind of stuff. And so I'm staring at spreadsheets. What the hell does this mean? But ultimately, that ended up being a 2012 paper that said, hey, the bladder's not sterile. The reason we bring it up is because this has been a really good relationship between a basic scientist stretching himself beyond what his comfort zone and a clinician who wants to do research, had done research, but not this type of research. I've realized that this very busy person, the only time she wasn't responding to my emails was when her hands were physically inside a patient. So any clinician that you need to spend 6 days waiting for a response is not somebody you want to work with. So you need to find the right people to work with. Okay? So,
thank you for sharing that. And although the two of us are standing up here today, there are so many more people involved in this story. First and foremost, patient participants who agreed to be part of our research by donating urine. Some of them, when they were asleep, as we passed the needle through their belly to get it. Before we did major surgery, so the needle wasn't too bad compared to the rest of the things we were doing to them. But people who had faith and were interested in moving this line of research forward. This really resonated with patients, and to this day, it's just not that hard to get urine samples from people. People are happy to do that. So now that we've figured out some of the details, I'll share some of that. We want to thank those patient participants and their families for their support, the funders who helped do this, including grateful patients who helped support us, and then many colleagues and collaborators and the research teams, support staff across This is an approved multitasking activity. If you haven't signed up for this, you get this nicely curated email from me every 2 weeks. You just go to JAMA Plus Women's Health, and then you'll see articles relative to women's health that you might not have otherwise seen. And if you don't like it, turn it off. But before you turn it off, write to me and tell me what I can do to make it better. So please sign up for JAMA Plus Women's Health. So I'm going to just talk a little bit about what I was trained with as a clinician. So you're going through medical school, you're going through residency, you're going through fellowship, you're learning technical things, diagnosis, treatment, side effects, management of complications. You're learning how to manage your time so you can take care of as many patients in a good way and get good outcomes as possible in a day without exhausting yourself. And learning how to do the technical aspects of procedures, vaginal surgery, laparoscopic surgery, robotic surgery. It's a lot to learn. And in every line of science and health investigation, there is a lot to learn. It's getting, and nobody can know it all, which is why we have colleagues and more reliance on things like AI, but whatever. So it's a busy time. So realistically, you don't have time to sit there in medical school and say, I'm not sure, let me see the evidence on it. Let's review for every single thing they're teaching you. Just as a matter of pragmatics, you gotta just take it as like, okay, it must be true, and you keep moving. And that's probably true for most of us in many things we do in life. So then you get into this pragmatic clinical care thing, you get taught that when a patient says this, or has these signs or symptoms, you do this, and this should happen. And if it doesn't, then you do this. And it's sort of this like road that you follow. And that works very well most of the time, even when it doesn't make sense. And part of the reason is patients have remarkable abilities to get better themselves. But for years, doctors like to take credit for this. But first they keep you waiting in an overcrowded waiting room, so I'm sorry about that, and I apologize for all my colleagues. But there is a way to begin to question these things and move the evidence, this pragmatic care forward. So I trained in OB-GYN, fellowship in urogynecology and reconstructive pelvic surgery, so my area was focused on the urinary disorders that were so common in the patient population that I was seeing. There's a whole other world. We have people who are dealing with men's health and the issues there and children's health. So there's a whole other world beyond what I took care of clinically. But so the one that I would see all the time would be urinary tract infections. And to this day, I can tell you I don't think we have that one fixed at all. And I'm going to focus on that one, not because I'm not aware of all the other ones, but because it is, I think, quite solvable if we put our minds to it. So clinical conditions, symptoms, and then all these dichotomies that we are taught, that you either do or do not have urinary tract infection. There's no spectrum here. Well, now that we know about the urobiome, that already sort of doesn't make sense. So one of the first things we need to do is moving beyond this dichotomy. And so some colleagues and I, through our NIH-funded, it's called the Prevention of Lower Urinary Tract Symptoms, we began to do some research in this area. And the first thing was to define bladder health. Now, doesn't that sound simple? Bladder health. What is bladder health, right? And it sounds like, okay, I know what bladder health is, and I know what UTI is, and I know, like, but if I push you and push you and push you very quickly, you don't. So one of the hardest conversations I have with medical students would be, so what's a UTI? And these are smart people. And some of them would roll out their very best, finely polished answers. Always wrong, always incomplete, always inherently illogical, inconsistent within itself. Because we actually can't define it. Same with bladder health. It's not just the absence of symptoms or the absence of a growth. And we learned some very interesting things as we began to develop the instruments to measure bladder health as a continuum as opposed to a dichotomy. Alan, I'm hoping you will jump in whenever I've left something out here, okay?
Yep.
All right. So I'm going to focus now on urinary tract infection. It is, but I submit to you it does not need to continue to be a problem. Alan and I had this nice little conversation about defining UTI. So this might have been a useful term when we didn't know so much. Like we used to say you have breast cancer or you have a heart attack. But if we just say you have breast cancer, that's fine for like the family Thanksgiving dinner or something like that. But if we're actually going to diagnose you or plan your treatment, stopping at breast cancer is nowhere near close enough. Saying if you had a heart attack is fine for like, you know, the news, breaking news at the bottom of your screen. So-and-so died of a heart attack. Okay, fine. But what kind of heart attack? Coronary artery spasm, atherosclerotic disease, aortic dissection. Like, there's some details we'd like to know as medical scientists, right? What else do you want to add?
Respiratory infections, right? So if it's a bacterium, they give antibiotics. If it's a viral respiratory infection, antibiotics aren't going to do any good. I'll bring up the breast cancer business again towards the end.
Yeah, just begin to play with that in your mind. And then context matters. Okay, you may have a certain situation now, but then if, I can't see you become pregnant because we have men in the room, but if a woman becomes pregnant and her obstetric team collects a urine specimen of urine culture and says, oh, you have an infection. She says, I do. What they really mean is your urine culture is positive. Okay. And then we have this completely goofy term that I'm highly allergic to, so please try not to say it around me, called asymptomatic bacteria. If you meet an asymptomatic pregnant woman, please call me, first of all. So asymptomatic during pregnancy is virtually unheard of. People have urinary frequency. A lot of things change when you're pregnant. Okay? In the urinary system, incontinence is common, urgency is common. So, and then bacteriuria. Well, if bacteria kind of live there normally, what on earth would asymptomatic bacteriuria mean? And why on earth would we expose a pregnant woman and her baby to an antibiotic that could alter that child's microbiome and have lifelong consequences? What testing should we use? No testing, very common. Eat these antibiotics, call me if you don't get better. No testing at all. Bad testing, a lot of that. Dipsticks, God knows when those sticks expired. You all know what dipsticks are, and you pee in a little blue cup, try not to get it on the thing, try to wipe it off before you give it to somebody, you know the drill. And then somebody dipsticks it and forgets that they're supposed to read it at 30, 60, 90, 120. Then we have the other layer of complicated or uncomplicated. This gets updated from time to time because you have very ill patients. Bone marrow suppression, bone marrow transplantation, they get a UTI, that could be a life-threatening event for them. Transplant patients, they get a UTI or a series of UTIs that could threaten the kidney that was just transplanted. So these are high-stakes things. We do not want real infections in these clinical scenarios. Okay. So then we have this term uncomplicated versus complicated, but it's not clear. I'm an English major. I was the token English major in my medical school class. So it's unclear what that refers to. Is it the patient who's complicated, i.e. I'm an immunosuppressed transplant patient? Is it the microbe who's complicated? Is the doctor complicated? We actually don't know, right? And so we have all these fantastic, beautiful documents where people come together, but this core thing is never addressed. And so we get guidelines, and you should do this, you should do this, but on an individual basis, a lot of it falls apart. And that complicated versus uncomplicated dichotomy, you gotta pick one or the other, drives treatment strategy. Self-care, very rampant. In the globe, people can buy antibiotic over the counter, not even from a medical person, so I shouldn't even say it's a counter. They can acquire powerful antibiotics. They can get ciprofloxacin, a couple doses on the street from somebody. And they just take what they, until they feel better, which is going to be about 24 hours. So if there was a serious thing going on, that few doses is probably not the right thing to be doing for a variety of reasons. It was probably more antibiotic than they needed. The duration was probably not right. Who knows? But we have a global problem with that. And then we talked about the lack of clarity, symptom relief, microbiologic cure. In every UTI antibiotic study, you'll see about 20% of the people don't get better. So 1 out of 5 don't get better. How would you like a treatment where 20% of you are still having the problem? Right? What if you would take a course and 20% of you are going to have to do it again no matter what grade you get? Forget this. That's crazy. Okay? But people relapse. So recurrent UTI is a serious problem that affects a subgroup of these people, typically in that group that didn't get better after their first one, but also in the 80% who did respond to treatment in the first, meaning their symptoms went away, they were able to resume normal life, 2 years go by, and then something else happens. But recurrent UTI is 2 and 6 months, some people say 3 and 12, so it's at least 2 and 6 months, is a problem, especially in postmenopausal women who are not using vaginal estrogen. And that first-line treatment of vaginal estrogen reduces that risk about 50%, yet many, many women are not appropriately treated as with genetics, but with other things as well. So basic things are not done. And then another whole area of UTI, especially with recurrent UTI, we have a lot of awareness now of microbiome. Out in the world, when people say, oh, that could affect your microbiome, they're typically meaning the gut microbiome. The gut microbiome kind of stole that word right away. Like, so we need modifiers. No, this is actually urinary microbiome. But the lay people want to have some ideas of, like all of us in our lay life, we want to be able to have like something that would help ourselves get better, help ourselves prevent recurrent infection. But we don't know how to help people recover from the antibiotics or from the dysbiosis of the infection itself, whatever that infection was. We don't know how to help that recover. So for UTIs, you're coming on your own. And that's really a problem when people are taking multiple courses of UTI antibiotics. Every 2 months, they're taking another 5 or 7 or 10 days. Or they're on a daily dose of antibiotics to suppress urinary tract infection, which is a treatment that people can use as well. So we don't know how to like help bring back the good players, the good lactobacillus, the things that can help augment their own defenses and their own system. So, yeah.
I'll talk about restoration following treatment later.
So we like to be able to predict and prevent occurrence of recurrent UTI if we could restore a healthy urobiome, which may look different for each person in this room. We don't know how to do that yet. The other thing I'd like to talk about is this gap between laboratory science to clinical care. And I think I wouldn't have invested as much time in my own personal professional life with Alan if I didn't respect laboratory scientists. I have great respect. Tremendous discoveries have been made there, and phenomenal work is being done there. But I have looked at 30 plus years of very elegant laboratory science. Mouse models, and this goes up to the kidney, and this happens, and this happens. See this colorful diagram, no reason. And I can tell you, in 2026, not a thing has changed in clinical care. Okay? Stop with the diagrams, be cautious with the mouse models, link tightly to a clinician so we can make meaningful change for the patients we're trying to treat. It's really important. Okay? I know the iron metabolism of this thing goes here, and that means stepping away from the ultra E. coli. E. coli is an important player and not the only player. Because when you get there, clinicians are notoriously slow for making changes. Because right now, this system is pretty easy for them. Hey, eat these antibiotics. gets bad, they send them to somebody else. But there's not many somebody else's, because nobody wants to take care of patients with recurrent UTI. Infectious disease doctors don't. They want sexy diseases like Ebola and cryptosporidiasis and God knows what else. But they do not want postmenopausal women with recurrent UTI. Urologists don't. They will cysto them, urodynamics them, and then send them back to you. So after they do the procedures that they can bill heavily for, they will send them back. And I'm speaking in generalities. OB-GYNs, my field, I don't think so. Just use more estrogen. They don't know. So there's very few people who really are invested in helping these patients resolve their clinical issues.
So all of the 30 or 40 years of uropathogenic E. coli research, okay, I'll talk about that term later, has taught us a lot of lessons about how certain bacteria, particularly certain E. coli's, can behave, right? But it has had, the only outcome is a vaccine that works on a handful of individuals. So it's been really, really great to learn more about, let's say, the circuit board inside of E. coli, and a little bit of Proteus and a few others. But it hasn't helped the clinicians one bit. So it's not a knock on Harry Mobley and Scott Holcren and their teams. They've given us a really strong foundation, but this generation has to go one step further. Learning more and more about less and less is not what you should be doing if you can. It's about thinking about the whole system, and I'll talk more about that a little later on.
Yeah. So I'm going to go a little bit more quickly now. So we have made progress, and I appreciate Alan raising this. There's been some really remarkable work, remarkable, beautiful diagrams, and the complexity of things has been increasingly recognized. But unfortunately, a lot of the progress has seemed to be equated with, well, let's find a new antibiotic, which isn't to me a solution. So I want to talk a little bit about the work that we did in the PLUS Consortium, and these three references help give how we collected urine samples in this cohort. And I'm happy to go into more detail, but for those of you who are interested, the bottom line comes on the next slide, which is going to be showing you the age of this group. We had about 435 unique individuals who were sequenced across these age groups, from 18 on all the way high. And this is the bottom line that you want to be looking at, this manuscript. And the reason is the sequences and the link for where you can find the sequences is in this paper that was published in 2025 in M Systems. So, and the reason is the data dictionary, the sequences, and now some additional work is also, I'll show you on the next slide, is available for you to work with through this effort. So you all know how to look through this, but this is the nearly 500 people that we collected urine samples on across the age group. And as with virtually every urinary microbiome study, they are not all the same. And this is not required, these participants were not recruited through a clinical cohort. These were not, they may have been patients, but they weren't anybody specific patients. These were just out in the community, and so you can see quite a variety of samples. Those samples now are being further analyzed by Lindsay Burnett, one of our colleagues at University of California, San Diego, who's working on the metabolomics of those samples and finding some pretty interesting things that actually might make a difference in clinical care. So I don't want to get too diverted over to metabolomics, but this link here on the top is where you can get that metabolomic data. And we have joined a small cohort of vaginal and stool samples that are linked with the urine samples for about 100 of these participants. So that could be of use for you. The advantage here is these participants are very well characterized with the bladder health spectrum, so that we know that they're not, it wasn't a good or bad. We can tell exactly where they were with a quantitative score along the spectrum, and they were measured multiple times. So the beauty of this is we figured out a way to get urine samples from these people. They can collect them at home. It's just as good as if they come into the research center. They can ship them through FedEx, which we learned a few odd things about that. For example, you cannot take the urine sample, put it in the tube, seal it, put it in the FedEx box. You cannot hand the sealed cardboard box containing a urine sample to a FedEx person. But you can put it in the FedEx box. Who knew? And so you ship it overnight and store it -80. The results are scientifically good and scientifically interpretable. So these are, I think all of us recognize, challenging times for science. We have a lot of clinical needs despite recognition of those needs. And the bottom line is we shouldn't diminish the patient's suffering. I mean, people don't go through all the hurdles to try to get an appointment with a clinician and all that kind of stuff if they're just like, and use the cost and try to figure out their insurance. They're suffering. They want help, please, please. So clinicians have a heavy burden. There's a lot of pressure on them to see and take care of a lot of patients in a short period of time. So they can't go spending 30 minutes to figure out a microgene report and call somebody and like see if Nas is free to talk them through it. It's not possible. So if you're going to try to make something that's useful for clinicians, it's really got to be with the patients in mind and something feasible that clinicians can do. We know that microbial resistance is an urgent global threat, and we need to address this with very, very high scientific standards. I'm looking at the people in the room that I know, and I'm sure we're in good hands with the high standards. So it really is up to you now to get started on your Nobel Prize, work in this area. I will try to live long enough so that I can see the Nobel Prize in relationship to the Urobiome, but before that, Alan's going to say a few things.
Right. So off of the idea of transitions, I'm going to have a whole series of terms and concepts that I think need to be retired. Okay? Asymptomatic bacteria. Well, what the heck does that mean if everybody, at least adult females, for sure, are bacteria? Right? Significant bacteria. There's plenty of evidence that the thresholds don't work. Okay? Basically, if somebody, if a clinician says you've got significant bacteria, what does it mean? I want to give you antibiotics. That's all it means. Okay? E. coli causes 70 to 90% of all UTIs. Now that's based upon the standard urine culture, or SUC, which I named standard urine culture because SUC sucks. Okay? It was designed for something entirely different, which was pyelonephritis by a very bright man by the name of Ed Cass. He was an IT doc at Harvard. He was trying to find a non-invasive way to determine whether someone had pyelonephritis so that no clinician did surgery on that individual while they had a kidney infection. It would then, transition to cystitis, to the bladder. And there's very little scientific evidence that the standard urine culture test works. Okay? It was designed to detect fast-growing bacteria like E. coli, right? That had no special nutrient requirements. It was non-fastidious. It was a facultative anaerobe, which means it doesn't really care what the atmospheric composition is, and it grows fast. Okay, so this is an example of the streetlight effect. That is, if you only look for lost keys under a lamppost, you begin to believe that keys are only lost under lampposts. This is a very, very old story. It actually goes back to recorded history to about the 12th and 13th century in Persia, where some important wise man was crawling around on the ground outside the mosque looking for something, and someone comes up to him and says, well, what are you doing? I'm looking for some important thing. He said, well, where'd you lose it? Well, in the mosque. So why are you out here looking for it here? Because the light's better out here. So the fact is that if you use an assay that detects E. coli, you're going to detect E. coli. So there are now more sensitive, more rational detection methods out there, and I'll show you a little bit of data later on. But the numbers probably about of the people who are diagnosed with, clinically diagnosed with a UTI, the number is about 40 to 50%. So E. coli is still a species that's associated in one way or another with urinary tract infection, but it isn't 70, 90%. Okay? Yeah, just so you know, I'm a little bit of an imp. I did name suck for a very good reason. And I find it just delightful that it's all over the literature. Yes. Okay. A specific pathogen is the cause of symptoms. This is a 19th century concept. Back in the day when we were trying to figure out what the cause of anthrax and cholera and tuberculosis were, it worked really, really well. But a clinician sees a patient and they have symptoms that are strikingly similar to other urinary tract infection episodes. And so, and maybe they do in fact send us a urine sample to the clinical lab and comes back positive for E. coli, and they say, I'm going to kill the E. coli because it's the cause. Except nobody does, because this postulates on any organism pulled out of any patient's bladder is suffering from UTI-like symptoms. So the best you can say is that E. coli is the suspected pathogen. But is it? Uropathogenic E. coli. Could somebody please define this thing? I can't. No one has, but the term is used all the time. What does it mean? As far as I can tell, it means it's an E. coli pulled from somebody's bladder or a midstream voided urine sample who is suffering from a urinary tract infection. And a non-UPEC strain, or an ASB strain, an asymptomatic bacteriuric strain, is an E. coli strain pulled from someone who wasn't having any symptoms. Okay? Well, if you look at their genomes, they're no different. And if you look at their transcriptomes, okay, so this is work from Jacob Hogans and Larry Reitzer from UT Dallas. All right, and so they did transcriptome analysis of a whole bunch of E. coli's, some from the E-Core collection, some that you'll see KE strains. That's their strains they isolated relatively recently. Up here is UTI-89 and CFT-73. We'll come back to them in a moment. But the UPEC strains are in red. The non-UPEC strains are in blue, and you can see that they do not cluster separately. Instead, they cluster by phylogroup, or essentially subspecies of E. coli. So you've got B2 strains, whether they're UPEC or non-UPEC, doesn't matter, they all cluster together. Their transcriptomes are similar. And the A and D strains cluster over here. So what the heck is a UPEC strain? I don't think, it can't be defined. All right, so UTI-89, CFT-73, these are the classic UPEC strains, along with NU14. There might be one or two others. Okay, so most of what we know about UPEC is from these two strains. All that really, really deep, very rigorous research on how UPEC works, how it behaves, is been done with those two strains. All right? What you'll find is they're outliers. They're not representative of what's circulating in the population today. Now, the E-Core strains were also collected decades ago. UTI-89 and CFT-73 were isolated about 20 or 30 years ago. But the E-Core strains at least co-isolate with the new, they co-cluster with newly isolated strains. So what we're learning when we do more CFT-73 and UTI-89, what we're doing is learning more and more about something that's not relevant to the clinical population. So please cut it out. Okay? How do I handle it? Because reviewers are not necessarily in this room. And they believe that these terms have meaning. And so how do I handle that? Well, I say that as a control, we're going to use UTI 89 to anchor it in the literature. That's the way I get around the problem of saying I don't want to do with UTI 89 because I don't want to learn about something that's not, and is no longer relevant. They're also lab adapted. I mean, they've been in people's labs for so long. God knows what they actually are. You're a pathogen. Okay. What's a pathogen? You got a colony on a plate. Is that a pathogen? A pathogen means a host. So how can you call even Bacillus anthracis on a plate? It's not a pathogen. Put it in a host, then it could cause pathogenesis. Although it won't always. There are plenty, I actually have done, believe it or not, anthrax work. I came by it sort of unfortunately, but no, my colleague died and I took over as medical. So I was interacting with scientists from Jordan and Pakistan who were doing anthrax work. And so if you look up my publications, it looks like there's 3 of me. But in any case, there were animals that had antibodies to anthrax, but had no symptoms. So is Bacillus anthracis a pathogen? Okay, so well, opportunistic pathogen? The heck is that? Right? So I'm now a disciple of these 2 people, Arturo Castelbel and Brodsky, who 2 decades ago started asking these questions and going, not everybody who was exposed to COVID got sick. Is COVID a pathogen? Well, in you it might have been, in me it wasn't. Right? Okay. So what we're really talking about are bacteria or viruses that have pathogenic potential. They could be pathogenic depending upon the ecosystem that they're in. You or your neighbor. Right? And so again, when I'm writing papers or grant proposals, I sort of try to say a bacterial species of pathogenic potentials takes up a lot of room, but somewhere in there I make the point that I'm trying to be very accurate with my language. And then I'll say you're a pathogen with it. Or I talk about a uropathogenic community, right? Which I'll get to in a moment. I suggest you read these papers. They're not very long, but they're very insightful. Arturo is very deep thinker. Okay, so our hypothesis is that UTI and other lower urinary tract symptoms are ecological problems, right? That they involve a prospective suspected pathogen or 2 or 3, right? The rest of the urobiome and the host protective system, right? Especially the immune system. Just because a suspected pathogen is detected doesn't mean that that suspected pathogen is the cause of the symptoms, or that there are even pathogenesis or symptoms going on, right? Asymptomatic bacteria. So here's an analogy. Thank you, Bob. It's a tale of 2 cities. Some cities are safe and some are not. Some people in this room might recognize this picture right here. Okay? The reason they're safe is that they have systems to keep for safety, right? All cities have criminals, but some are under control because there's a police force, there's a political system that works, there's neighborhood watches, there are all sorts, there's structure, okay? And then there's cities where the criminals are in control. So the urinary tract isn't any different, right? There's some communities of microbes that are uroprotective and some that are uropathogenic. So the question is how can we tell them apart? Okay, a little aside, shadow divers. I was reading this book, which has nothing to do with microbiology, when Linda and I were talking. It's about deep sea divers who discovered a U-boat from World War II in a location off the coast of North Carolina that wasn't supposed to be there. And so they did a lot of work in the archives, and then they went down as far as they could go, and they were trying to identify what U-boat it was. And ultimately, one of these guys had to take his air tank off and pass it through a narrow passage to get into the engine room that was all twisted up to pry off the one thing in that submarine that would identify its ID number. Okay? Now, you go down that deep, and you've got about 30 minutes to be able to work down there because it's going to take you hours to get back up so you don't get the bends. That's one problem. The second problem is that, well, if you're not careful, you can die down there. And two of their team did, a father and a son. You get confused, and then you make mistakes, and then you die. The other thing is that you've seen pictures of crystal clear waters or animals that no one's ever seen before, and they're really crystal clear. That's because there's this huge bright light, and nobody's moving. But at that depth, there's no light, unless it's artificial, and the moment you move, it gets silted. And so these deep sea divers work by touch, intuition, and experience, because they can't use their eyes. Clinicians. I called her a shadow diver because she was working on very little information. Okay? That they were the best clinicians were the ones with the best instincts based on the best experience. Okay? So what do clinicians make decisions on? Very little information, dipstick, suck, and experience. A few other things. Okay. The typical clinical approach is dipstick with reflex to suck. So if the dipstick says maybe positive, then send it a urine culture off to the clinic. That's what reflex to suck means. But they're both deeply flawed. Right? First of all, the dipstick depends upon, to a large degree, two things. Okay? One is their nitrate reductase activity, and two are their leukocytes, white blood cells. Well, many species, and I'm going to show this to you, okay, don't have the gene for nitrate reductase, so they don't have that activity. Okay? Number two, leukocytes are fragile. By the time it gets red, okay, many of those white blood cells have broken up and died. Okay? And I already told you that suck is biased. So here's a table put together by my colleague here, Bob Moreland. And you've got, this was a study of 823 individuals that had been diagnosed with UTI using a multiplex PCR panel by the people associated with the diagnostic company Diagnostics. And I have a conflict of interest. I'm on their scientific advisory board. Anyway, this is the order of the prevalence of any given organism. So E. coli up here was detected 41% of the time. And then here's Enterococcus at 22% of the time. And then down here's Klebsiella, 13% of the time. Okay. By the way, this number 22 is wrong, it's 27. Okay, so suck detects E. coli, no problem. This is designed to do that. Detects Klebsiella, no problem. Suck underestimates Enterococcus faecalis. We've reported on that multiple times. It cannot detect Actinotigrum shallii or any other of the Actinotigrum species. It very rarely detects Streptococcus aeruginosus and very rarely detects the Aerococcus urinae complex. And yet, these are the most common species detected in people that have UTI, and these are bacteria that have pathogenic potential. But only E. coli and Klebsiella of the set have nitrate reductase. So you come in with symptoms. You have no E. coli, you have no Klebsiella, no Pseudomonas, no Citrobacter, no Morganella, none of the enterobacteria, right, that have nitrate reductase. And so you are, your report comes back no growth. And the dip is negative, right? So how can you tell the healthy from the unhealthy communities apart? Okay. Well, let's look at the host response. These are 3 different immune markers that pathognostics discovers that would allow them to predict whether someone actually had been diagnosed with UTI. NGAL, IL-8, and IL-1 beta. And as you can see, the percentage of individuals that have been detected with E. coli, the numbers here for E. coli are no different than they are for these very positives, Aerococcus actinotigna, the variance group strep, which we now know is almost exclusively strep anginosus in the urinary tract. Well, actually, a new species called Streptococcus vaginalis. Okay, and here's another way of looking at it. It's the same data. So here's E. coli and the 2 Aerococcus actinotigna. Just so you know, here is Canada, here's GBS, Morganella, Pseudomonas is up here. I mean, if you're studying these bugs here, you're studying the wrong bugs. They're just not very prevalent in individuals that have urinary tract infections. These are the bugs to be studying, okay? Somewhere along here is your cutoff. Every one of them had an immune response that was pretty similar. Well, they only had 2 of them. So 1 out of 2. Okay? So Aerococcus actinotigna, the strep anginosus group, and they're not detected, and nobody knows what to do about them, but they are bacteria with pathogenic potential. Okay? Towards the end. How do we push unhealthy communities, dysbiotic ones, towards healthier eubiotic ones? Okay. I'm going to use another analogy. I live on the prairie. I live in the northwest suburbs of Chicago, and that literally is the prairie. Within a mile or 2 or 3 of my house, I have both cornfields and prairie. And there's something called forest preserve in the Chicago area, so there's a lot of green space, and a half mile of my house is one of those forest preserves. And there was a cornfield at the far end of it that the Park District bought, tore down the cornfield, and restored wetlands and prairie. Now, if they just had torn down the cornfield, think antibiotics, this is what they would have gotten. Because I know I did that in my yard. During the pandemic, I decided to tackle the jungle that I'd let my yard become because I was working in the lab 7 days a week for the last 400 years. Okay, so after tearing down the trees that I could and bringing in an arborist to bring down the trees that never should have gotten that big or planted where they were, I had a lot of dirt. And my yard looked an awful lot like this. Well, actually it didn't, because they did a lot of weeding. But that's what would happen. And there was a part of my yard that actually let the prairie grow in, because it's not very far away. But it wasn't really prairie. It was whatever landed there. So if you want to do this, and this thing is about 3 or 4 miles from my house, you need to know what a prairie in that particular neighborhood is supposed to look like. And then you purposely plant those plants, and you restore the earth, the soil. You got to do that, or you don't get prairie. Okay? So another analogy is regenerative farming, where you actually restore the earth, the soil. You use animals and different types of crops. So this is regenerative. This is not. Okay? So here's an example of an attempt to do regenerative healing. So this is a recent paper from Susan Gross group at MedStar. She's a collaborator of mine, where they're actually using lactobacillus. Well, it's no longer lactobacillus rhamnosus, but it's lactobacilli bacillus rhamnosus. One of the dangers of whole genome sequencing is that everybody gets a new name, and we have to keep up with it. But what they've been doing is putting LGG, which is a live biotheric product or probiotic, into the neurogenic bladders of spinal cord injury patients and getting positive results. Okay? So that's an attempt to do restorative work. It's only going to work to some degree because it's only one bacterium. We probably really do need a consortium. We don't know what that consortium is. And so what we have to be able to do is distinguish between healthy and unhealthy microbiomes. We need to identify and sort ecosystems. Just as the people who study breast cancer figured out that there were 4 major categories, right? Luminal A, luminal B, HER2 positive, and triple negative. And if you get triple negative, there are not a whole lot of treatments for you. Okay? But the others, most of those people get better. Why? Because of molecular detection methods, which many clinicians, I've heard a couple of clinicians say that molecular detection methods are all predatory. Just a recent meeting, and I'm like, what? Some are, some are not, right? We need to know what's there, and we need to get clinicians to help us collect all that information, sort them into categories, and with AI and other machine learning type of approaches, if we have enough data, we can sort it all out, okay? But then we need clinicians to embrace the knowledge and figure out what treatment algorithms with new treatment options. Some of you are working on that, right? So that if you're in ecosystem 1, well, out pops, from the algorithm, out pops, do treatment 1. Like we do for breast cancer. Doesn't mean you're going to get better, but you have a better chance of getting better if you do. So embrace the new knowledge, and that's the way to improve patient outcomes. And as Linda said, we want to thank the attendings, the fellows, the residents, nurses. By the way, for those who don't know, the attendings are the senior doctors, right? My lab members, our collaborators, many of you are out in the audience, funders, and of course, the study participants.